You learned about dihybrid crosses in the last video last week. And in a dihybrid cross, we assume that the two genes of interest are on two completely separate chromosomes. So here we have a pair of homologous chromosomes and another pair of homologous chromosomes. Maybe this one's from dad, this one's from mom, this one's from dad, this one's from mom. And we are assuming that these two genes are going to move independently of each other. So that means that this big P could possibly move with this big L, or this big P can move with this little L. And similarly, this little p can move with this big L, or it can move with this little l. That's how we come up with these gametes right here. And that's how we fill out our dihybrid cross. However, what if we knew that the genes were really close together? What if the genes resided on the same chromosome? If they resided on the same chromosome, that's definitely going to impact how they move. And so we wouldn't necessarily see the typical dihybrid cross that we were speaking about last week, even though we're talking about two genes here. This is called linkage. And as you can see in this picture, the genes with the alleles P, big P, little p, or big L, little l, reside really close together. And because they reside close together, that is going to impact how they move. And as you can see in this picture, the big P only moves with this big L, and this big P only moves with this big L. This little p only moves with this little l, this little p only moves with this little l. So that will impact dihybrid crosses and affect the pattern of inheritance. So linkage is when two genes are on the same chromosome. Because they're on the same chromosome, they are not going to assort independently, and so you can't do a typical dihybrid cross. You would have to link them together, which is why they're called linkage, right? So the big P is going to move with the big L, the little p is going to move with the little l, and that's how it would look. So try and think about what mechanisms we've learned that can actually screw this up. What can possibly disrupt this? If you can remember back from meiosis, learning about the mechanisms that contribute to genetic diversity, crossing over can impact how these genes are going to move, because they are going to make new recombinations of these genes in their gametes. So crossing over can impact this, and there's actually a number that you can calculate called the recombination frequency, which means how likely are they to recombine or not. If the number is really small, like 5%, they are less likely to recombine. And what that means is that they are going to reside really closely together because they're less likely to recombine. So this would be an example of like a 5%. If the number is big, like 60%, you can assume that it would be 60% more likely that recombination would occur. So maybe the genes are actually pretty far apart, because it's likely that recombination can possibly occur here. So we're not going to do this in our class, but it is an interesting way, because you can actually map out where different genes are on the chromosome using this number recombination frequency. Mitochondrial genes. Mitochondria have DNA of their own. Mitochondria, there is a theory that mitochondria used to be prokaryotes on their own, and our eukaryotic cells ingested them somehow, and they became a part of our cellular organism. And the reasoning behind this is that, one, we know that they produce their own energy. Two, they carry DNA of their own, just like bacteria do, just like other prokaryotes do. All of our mitochondrial DNA, all come from our mother. And so that's actually pretty cool that all of your DNA comes from your mother. It's going to encode for RNA, and then that's going to code for protein, which is going to produce energy. So all of our mitochondrial DNA always all comes from our mother. So this is showing you the pattern of inheritance of mitochondrial genes. So let's take a look from the top. The first generation here, this is a female, an affected female. And one thing you can notice is all of her children are affected. And that's because all of her children all receive her mitochondrial DNA. They're all going to be affected. Let's look at the second generation and the third generation now. In this first family over here, this person is a male. Notice none of his children are affected. This, on the very far right, this person is also a male. None of his children are affected. This middle, none of his children are affected. The middle family is here with the affected female. All of her children are affected. And this one over here, all of her children are affected. And that's because these children received mitochondrial DNA from their mother. Same over here, they received the mitochondrial DNA from their mother. They're not affected. So what kind of diseases does mitochondrial DNA impact? They're going to impact tissues that have a lot of mitochondria. So tissues that are going to produce a lot of ATP. And maybe you can guess that that would be muscle. And so a lot of muscle degenerative disorders have to do with mitochondrial DNA. So some symptoms can be fatigue, flaccid muscles, intolerance to exercise, or ragged looking muscle fibers. Okay, continuing on to our complexities of heredity here. We are talking about polygenic traits. Poly means many, right? Like a polygon. So polygenic just means many genes. These are traits that are impacted by many genes, multiple genes. A good example of this is eye color. Eye color is dictated by many genes, at least 4, more than that though, over 4 genes contribute to eye color. Some have larger contributions than others, and this is why we have such a rainbow of eye color. Is because that there's just more than one gene, which means more than 2 possible alleles. Polygenic traits are when there are multiple genes affecting the trait. Multifactorial traits are when traits are polygenic, but also when they're affected by the environment. So it has a genetic component, a lot of genes, and then it has environmental factors affecting it too. An example of this would be skin color. So skin color is a multifactorial trait. First off, it is dictated by more than one gene. It's dictated by many genes. And then our environment is going to impact that. For example, if you like to sunbathe all day, every day, that's definitely going to impact your skin color versus somebody else who prefers to stay inside. So a good example to observe this is to look at plant height in peas. This is a single gene trait, so it's not multifactorial, but it does have environment involved here. And so it's a single gene trait, and we're just doing a single gene for sake of clarity here. It's just easier. These are going to be tall plants versus dwarf plants. If we look at this graph, over here we have plant height. The higher it goes, the higher the plant goes. Over here we have amount of water. So that means over here it's very little water given. Over here it's a lot of water given. And what we're trying to say is the genetics are going to have an impact. But notice, if both of these plants didn't get that much water, they're both not going to be that tall, right? They'll be a little bit different, but they're still going to be relatively lower height. So the environment does impact here. You'd have to have the proper environment to make sure that the plant is growing at its best height or at its strongest. And then we can see the full capacity of the phenotype, where you have a dwarf plant here given plenty of water and a tall plant here given plenty of water. Plant height in peas is considered a discontinuous trait. And what that means is that there is no in between, right? The plants are either tall or they're dwarf. There is no dwarf and then a little higher than dwarf and then a little higher and then tall plant. So there's only two distinct phenotypes. So an example of this is on the left right here. We've got a dwarf plant, and a tall plant. And this is the frequency of individuals, meaning what they would have their babies, what their progeny would be. And so let's just say this is lowercase d, uppercase D, uppercase D. This is the parental generation. If they were to mate, this would be the F1 generation. Notice that they are all tall. And if we were to cross this, what we'd get is 75% is going to be tall, right? They could either be big D, or big D, little d. And then 25% are going to be dwarf. This is what we've been talking about. All of those Mendelian inheritance Punnett squares have been discontinuous traits. But we're talking about complex traits here. And what a continuous trait is what if there were in-betweens? There was a middle one and another middle guy and another middle guy and another middle guy. That's what a continuous trait is. And so here, say we have little d, big D. These are both big D, little d. But because we have some continuousness, these are going to be different based off of their genotype. They are going to give many phenotypes in a range. This is just an example, and it's super simplified right here, but we're going to get into a little more detail. Human height is a great example of a continuous trait. So that means that all the heights in a specific range are possible. And human height is a multifactorial trait. It is dictated by multiple genes, and it's also dictated by environment. A good diet, exercise, proper amounts of sleep. Those are all really important to make sure that a human being is growing as they should be. Human height is an example of a multifactorial trait, and multifactorial traits are usually continuous, and so you're going to have a range. So take a look at this picture right here, and this is, I think it's a high school, and it just shows the frequency of heights, right? And so at the lowest and the highest heights, there's a small amount of people, a smaller amount. And then as you get into the intermediate, there are more. So this means number of people, right? So the phenotypes of multifactorial traits usually form a bell-shaped curve. So let's say, for example, that this phenotype, this trait, height, human height, is controlled by two genes, and let's just call them A and B for now. If you have a big A or a big B, that's going to make you taller. If you have a little a or a little b, that's going to make you less tall. And this is incomplete dominance, right? Because we're having a blend of phenotypes here. So if we continue on with this example of little a, little b being less tall, and then big A, big A's and big B's being more tall, let's also assume if somebody has the genotype little a, little b, they have a height of 5 feet tall. Every dominant allele adds 6 inches. And so if they have one dominant allele, that's going to add 6 inches, that would be 5'6". If you added another one, that would be 6 feet tall. If you added another one, so you have 3, that would be 6'5". And then if you added another one, that would be 1 foot, 2 foot, 2 feet over 7 or 5 feet, right? So that would be 7 feet. So assume big A, big B, little b mates with another person who is big A, big B, little b. What would their kids look like? And you have to make a dihybrid cross here. There is no linkage. And what you would see is this sort of graph. The possibility of having no dominant alleles is less frequent. The possibility of having all dominant alleles is less frequent. What's most frequent would be to have 2 dominant alleles. Having 3 would be slightly less frequent than that. Having 1 would be slightly less frequent than that. And so what we're trying to show you is that there's that bell curve. Our simple example of using only 2 genes has 5 different categories of height. But what if height is actually affected by 3 genes, A, B, and C? How many categories would we have now? You would see something like this. So you'd have 1, 2, 3, 4, 5, 6, 7. 7 categories. And again, notice it makes a bell curve. If you have 4, you have 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. And so notice that even though we're adding the number of genes that could possibly dictate this one trait, it is still forming this bell curve. Now you add in the environment, nutrition, age, exercise, injury, stress, and the trait is continuous.